Good morning to all of the audience. On this day, in this morning, I am glad to be one of many researchers and young scientists to be able to attend the 15th Asian Congress on Biotechnology in conjunction with the 7th International Symposium on Biomedical Engineering with the topic Biotechnology for Stronger Recovery. In this opportunity, I am Urbanus Yustus Lebuan together with Ms. Roga Florida Kembaren, Ms. Meryam Martgrita, and Mrs. Rizlani Kholibrina proudly present our research paper with the title Thrombolytic Protease Characterization from Leaves and Fruit Flies of Jernang Rattan plant or Daemonorops draco. As the outline, what will we discuss will be divided into 4 points. The first is introduction, followed by methodology, results, and discussion, and will be finished with conclusions. What became our background in our research paper is that in the last 5 years, thromboembolic disease is a major problem that is very harm to human life and health. The need for synthetically produced drugs still has drawbacks in its use. For example, dependence on drugs in high dosage because of the short half-time. On the other side, natural drugs that has been produced are still limited in the amount. In fact, developed countries rely heavily on natural or herbal medicines, but out of 362,000 plant species in over the world, only 10% have been known to be used as medicines. One of the plants that we focus on is Jernang rattan plant or Daemonorops draco. This rattan plant type is widely found in Indonesia, especially in Sumatra Island, which is generally used in the commercial aspect because of the high economic value of the resin. In addition to its usefulness from a commercial aspect, turns out that in health and medical aspect, Jernang rattan plant resin has been used as anti-tumor, antitoxic, antimicrobes, and anti-thrombosis agents. As we know, the use of this Jernang plant resin itself is the same as the activity of protease enzyme in the treatment of thrombosis, where the protease enzyme will degrade the blood clot that form in the blood vessels. So, ladies and gentlemen, as the goal of biotechnology for stronger recovery, in today's Congress, what become our objectives is we able to utilize the leaves and fruit flies of Jernang rattan plant as the local plant to produce the biomedical agents to identify and characterize its protease activity. Audiences, our research methodology are divided into 4 main steps. The preparation step had done to get a clean and fresh raw material. It is the leaves and the fruit flies of Jernang rattan plant. The specimen we used then stored in the freezer. Protease sample preparation were done in 3 procedures. The leaves and fruit flies of Jernang rattan plant will be extracted separately as the variations. The crude extract then measured to get the total protein content with the Bradford reagent. The extract produced then enter the ammonium sulfate precipitation process with fraction variation used is 20, 40, and 60%, followed by 1 night-long dialysis step to obtain the protease enzyme. Crude protease enzyme will be characterized in the 3rd step. We had done the casinotic assay in the agarose casein media to identify the protease activity in the sample. Also, we had done the fibrinogenetic assay together with the human fibrinogen sample to find out how was the protease activity results in the electrophoresis gel. And finally, we had done the Follin-Culture assay to measure the amount of protease enzyme that contained in the sample based on the L-tyrosine substance released in the casein sample. In the 4th step, we had done the inhibitor assay procedure to characterize our protease enzyme sample based on the inhibitor that works to inhibit the sample activity. We use 3 types of protease inhibitor. It is phenyl methylsulfonyl fluoride as the serine protease inhibitor, ethylene diamina tetraacetic acid, and ectasic acid, both as metalloprotease inhibitor. Ladies and gentlemen, now we have arrived in result and discussion. From the total protein content measurement with Bradford region, it's shown that in the graphic in the slide that the fruit flesh of Jernang rattan plant specimen contains more protein than the leaf specimen, which reached almost 0.0002 milligram protein from 1 milligram of fruit flesh of Jernang rattan plant. As the ammonium sulfate procedure were done, protein concentration in the sample rises as the ammonium sulfate fraction added. From both of the specimen used, we were able to obtain the highest protein concentration in the 60% of ammonium sulfate fraction variation. With the sample result, we used it to continue the next procedure. In the next procedure, we had done the radial casinovic assay in the agarose casein media. As you can see from the picture here, the clear zone produced from both of the enzyme source increases as the ammonium sulfate fraction. At 60% of ammonium sulfate fraction, the leaf sample able to create 1.4 cm diameter of clear zone. With the same fraction used, the fruit flesh sample able to create 1.8 cm diameter of clear zone. This result indicates that the sample have the casinolytic activity. By using Follinsio-Corti assay, we were able to know that the amount of enzyme added in the leaf sample, 60% fraction, is 1.58 microgram, while amount of enzyme added in the fruit flesh sample, 60% fraction, is 0.67 micrograms. As a comparison, to find out which specimen has more mass activity, the sample from fruit flesh specimen then diluted until the same amount of enzyme, and then we redo the test. The result is as shown in this picture. Leaf specimen sample creates clear zone wider than the fruit flesh specimen, which means that the protease activity from the leaf extract works better. Audiences, as we mentioned before, that Follinsio-Corti assay had done as the quantitative enzyme activity measurement. This step's goal is to determine the enzyme activity based on L-tyrosine substance that released in the mixture. In the slide, we can see that the protease enzyme activity produced from fruit flesh of Jernang rattan plant has the greater activity with 0.17 units per milliliter sample. Qualitatively, protease enzyme activity were performed with the presence of human fibrinogen sample. This procedure goals is to show the activity of protease enzyme from both of the sample to human fibrinogen presence. Electrophoresis gel results show that the leaf specimen enzyme have degraded the alpha chain of human fibrinogen in 240 minutes of incubation and able to degrade the gamma chain during first mixture, while the beta chain were unable to degrade by the enzyme sample. At the other results, fruit flesh specimen enzyme able to degrade alpha chain only in 180 minutes of incubation, also able to degrade the beta chain in 60 minutes incubation, and degraded the gamma chain in the first mixture. So we can conclude that enzyme sample have the protease activity to human fibrinogen. As the last procedure, we had done the protease enzyme inhibitor assay with 3 types of inhibitor. This procedure's goal is to determine the type of protease enzyme from the leaves and fruit flesh of Jernang rattan plant based on the inhibitor substance that reduce its activity the most. The results show that phenylmethylsulfonyl fluoride inhibits both of the specimen enzyme the most with 11% residual activity of the enzyme. From this result, we can conclude that the protease enzyme from both leaves and fruit flesh of Jernang rattan plant specimen will belong into serine protease. Ladies and gentlemen, we now have arrived in the conclusion. Based on the result from our research, we can conclude that first, protease enzyme isolated from the leaves and fruit flesh of Jernang rattan plant have the highest activity at 60% of ammonium sulfate fraction. Second, protease enzyme isolated from the fruit flesh of Jernang rattan plant able to hydrolyze the alpha, beta, and gamma chain from human fibrinogen. And the third, protease enzyme isolated from the leaves and fruit flesh of Jernang rattan plant are belong to serine protease. Our conclusions will end our presentation. Thank you for the time and attention had given to us.